You can ask questions over the recording, of course. Okay, first thing we have to do is let's get to the material. Oh God, I can move the cursor, but I'm frozen. So I got to get out of this. I can't even stop share. Hmm. There it goes. Okay, are you with me in the center of the screen here?
Yes.
Yes.
Okay,
Yes.
we're all set to go. I skipped a lecture yesterday to give us more time, and it was the longest session we've had so far. More time to look at the bones, with the exception of the skull. We'll do the skull today. That won't take as long as the other bones did, so we need to get into a lecture. The next lecture in order, although it was supposed to be yesterday, is cell division, which I consider the last of the remedial topics, because muscle tissue comes in then on Monday, which is a good spot for it. Okay, that's my phone. I'm sorry, but I don't even answer it. It just annoying. That's a spam call. Spam call. Where's it from? It's from Superior, Arizona. I don't know anybody in Arizona except those idiots. Okay. Okay, so we're going to look at cell division. And I know for most of you, this is a rehash, but please don't assume that because you had it somewhere before, and I know you did, that you still remember all of it. If you do that, you're going to be missing a lot of questions later on. Okay, I'm going to get rid of you guys for a second. Okay, this first part is definitely remedial, I would have to call it. Okay, but we're asked to do this. Uh, Sometimes I don't want to do something, but I have to go along with the course descriptor. And yes, when we get to histology, knowing this material is vital. Okay, so cell division, there's just two types, mitosis and meiosis. Sometimes mitosis is called somatic cell division. You don't hear it very often. Somatic means of the body. It's division of cells that constitute replacement, replenishment, or growth of the body. Whereas meiosis is referred to as reduction division because the number of chromosomes in the cell is reduced by half to form the gametes or sex cells. So the mitosis would take place body wide. Meiosis would only take place in the sex organs or of the ovary and the testes. We'll do mitosis first. It's always a bit easier to get into. If you understand the consequences of mitosis, you've got half the battle. It's one diploid cell. The term diploid is abbreviated 2N. The term diploid means that the cell has two complete sets of chromosomes. Okay, so think of any cell in your body that's alive still. Don't think of a red blood cell. It doesn't have a nucleus. It doesn't have this. It's on its way out. Don't think of the surface of your skin. It's all dead and it's on its way out. But think of a liver cell, for instance, or a stomach lining cell, anything else in the body. You have two sets of chromosomes. One set you got from your mother, and one set you got from your father. In order to form a division where one cell divides by mitosis to form two cells that are each going to be diploid. In other words, we have one diploid cell, might as well say it, that has 46 chromosomes in it. And you divide and form two cells, and those will each have 46 chromosomes in them. Where do we get the extra chromosomal material? Obviously, it has to be manufactured within the cell prior to the division itself. Between divisions, while the cell is not dividing, it's functioning, it's doing what it's supposed to do. The liver is cleansing the blood. The lining of the stomach, for instance, is absorbing material. But these cells are not in the division cycle yet. Between divisions, the process is called, or the situation is called interphase. Interphase between divisions. Since the cell is working hard during interphase, and even during interphase, it's also producing the extra DNA it's going to need when it divides. This is not an inactive phase. I think when I was first taught about interphase, I was told it was the resting phase. That's ridiculous. That's absolutely ridiculous. The interphase cell, or the cell in interphase, is really, really quite active. In humans, the nucleus of normal somatic cells, cells of the body, contain 23 pairs of chromosomes. The total number is 46, as we said a moment ago. You get 23 chromosomes from your father and 23 from your mother. If you think, for instance, of a deck of cards, and let's just pull out all of the hearts and all of the spades. Okay. And I don't care if your spade is high or low, it doesn't matter. We've got matching cards. No jokers in this deck, by the way. We've got two sets. How many sets are there in a deck of cards? And that's why we don't use it as a good example. There's four different suits, as we call it. But a set of chromosomes varies from different organisms to different organisms. In humans, a set of chromosomes is 23, and that's called the ploidy number. Okay. When you combine the 23 chromosomes from your father with the 23 from your mother, you'll get 46. And you'll not have just one N, you'll have two N. Okay. 2N is what we call diploid again. Okay, so the number of chromosomes that make up a complete set is the ploidy number. I don't know any other animal that has 23 as its ploidy number and 46 as its total, but we're not here to discuss the other animals anyway. Okay. Here are the stages of mitosis. Okay. Remember, before the stages of mitosis or cell division begin, interphase has been going on. The cell has been functioning and it's produced extra DNA so that when it splits, half of that extra DNA can go to one cell and half to the other cell. I don't know of any division that doesn't result in the formation of two cells. In other words, one cell doesn't split to become three. One cell splits to become two, even in meiosis, as we'll see in a moment. The first difference between the interphase cell, and that's the one we looked at last week, the first difference that we notice when it begins to actually divide actually are these four things here. The nucleolus, which was the source of transfer RNA. Told you didn't have to remember that. Okay. The nucleolus disappears on us. It's not disintegrating. That's really a bad word, and I've used it a little further on. But we don't see it anymore. It becomes less dense. The chromatin, that's the genetic material dispersed through the nucleoplasm, starts to condense and become visible chromosomes. We don't see the chromosomes with the microscope in the interphase cell, but they start to appear in prophase. The chromatin is tightening up like that plastic rope I talked to you about the other day, tightening up to protect all of its surfaces. The nuclear membrane itself begins to disintegrate. I wish probably I hadn't used that word. It begins to disappear. We don't know where. It probably is a continuation of the endoplasmic reticulum and goes out into it. But no matter what, under the microscope, the nuclear membrane is no longer completely visible. And then the centrioles that we talked about, okay, these will finish splitting and they migrate towards their poles. North Pole, South Pole, no, that's not really good. Just opposite each other on opposite sides of the nuclear area. Okay, so these things happen. Nucleolus disappears, chromatin condenses to form chromosomes. The nuclear membrane begins to disappear, and the centrioles split and migrate towards poles. Okay. And I have a diagram for you for this in a moment. Metaphase. The spindle apparatus has been set up. What does that mean? Well, the two centrioles that are going to be on opposite sides of the nuclear area will send out spindle fibers. These spindle fibers attach to the center of the chromosomes that are now becoming visible. The chromosomes align on the equator of the cell, the midline of the cell. The chromosomes do not need to pair off at this stage, in case you're thinking of meiosis. The 46 chromosomes only need to be at the midline. They are attached by a spindle fiber to a centriole on both sides. It's almost like a fish in the water, and somebody on a tuna boat, that's the centriole, has cast a line and hooked a chromosome. But on another boat, the other centriole, a line is cast and it hooks it from the other side. We'll see what this does in a moment. I don't want you to worry about understanding it yet. You will in just a second. And by this time, the nuclear membrane has completely disappeared on us. The chromosomes will split now. And it's the centromere of the chromosome that does the splitting. Think of a piece of yarn or a string, okay? Just one piece of string. Give it a color in your own mind, okay? Now, tie a knot in the very center. So instead of just a piece of string, you have string, a bump, that's the knot, and the rest on the other side. The string itself is called a chromatid, and the bump is called a centromere. Okay. It's also known as a kinetochore, but that term came and went years ago. You may still find it. We're always going to use the word centromere. Okay. Now, if you have it in mind, you have a piece of string with a knot in the center. That represents one chromosome. That chromosome can duplicate itself. So I want you to take in your mind a second piece of string, the same length, the same color, and tie it in the center so that the center is a slightly bigger knot and 4 strands are coming away from that center. Well, what's happened is the chromosome, the chromatid is a single strand. In this case, the chromatid started off as a chromosome. It doubled. It's able to reproduce itself. The genetic makeup, sequence, everything's exactly the same. Both of the new strands are identical. There is one centromere holding 2 chromatids together. The number of chromosomes you have is equal to the number of centromeres. So in our example, even though there are 4 strands because there are 2 chromatids grouped together, we only have 1 chromosome. Looking under a microscope, or you've seen it on the forensic shows, looking for DNA and such, the centromere represents the middle portion, or sometimes the end portion, but a portion where the chromatids are bound together, and the centromere equals the number of chromosomes. If there's only one centromere, only one chromosome. Well, in our cell that we're talking about here, there are 46 chromosomes, and they're already doubled. In other words, they have 2 chromatids apiece. They did that in interphase. They did that in preparation of splitting and half going one way, half going the other, half being trapped in one new cell, half in the other new cell. So 46 chromosomes, but twice as many chromatids. If there are 46 chromosomes, let me ask you this. If there are 46 chromosomes now lining up, taking their positions to divide, but still there's 46 of them, how many centromeres are there?
23.
No,
46.
46. Okay, the number of centromeres always equals the number of chromosomes. Okay. Now, we'll come back to that in a second. Telophase After the chromosome.
Charlie,
Yes.
before we move on, is that true in every stage of mitosis that the number of centromeres always equals the number of chromosomes?
Yes, it is.
Thank you.
Always true. Okay. It's very rare that I want to use the word always. There's always an exception, it seems, but there isn't here. There isn't here. Okay. Now, as I said a moment ago, picture just picture one chromosome for a second with two chromatids. It'll have only one centromere, and it's been hooked by these two fishing vessels, which we know as centrioles, and they're pulling on it. They're pulling on it from both sides. And what happens is the centromere splits. The centromere splits in two, each part of the centromere taking with it towards a centriole, taking with it a chromatid. So we had two chromatids bound together by one centromere a moment ago, and now we have two chromatids, but they're separating from each other. And how many centromeres do we have? Don't guess. We have two. Because when we break the centromere by pulling on it from both sides in a tug of war, like I was mentioning, each side, each piece has the ability to regenerate the other half. And we consider each piece to be a centromere. Okay. So when the 46 chromosomes all split, or they're all split by the spindle fibers, and they're pulled towards the centriole, for a moment, there aren't 46 chromosomes. You pull them each apart and make two out of one, and there will be 92 chromosomes. That's represented by 2n plus 2n. And it's a very temporary situation. Okay. The cell isn't functioning at this time to do anything except divide. We'll come right back to that. The half of the chromosomes go to one centriole area, and a nucleus reforms around it. The other half go to the other centriole area, and just outside of it, a nuclear area reforms around it. Okay, here's what we actually said. And I know sometimes it's better to do the diagram first, but this forces you, I think, to think a little. And okay, here's interphase. Interphase is not the resting phase, it's the active phase of the cell, actually. And it's like our drawing the other day. It doesn't look like it, but it's the same. Here's the nucleolus. Here's the nucleoplasm that has the chromatin in it. Here's the nuclear membrane. Out in the cytoplasm, the only thing drawn in this drawing is the centrioles. They have been shown here as already having split. The number of chromosomes we have inside is 46. 23 came from your mother, 23 came from your father. Your mother's chromosomes have only your mother's genes. Your father's chromosomes have only your father's genes. So you have your mother's and father's genes for the ACE, for the 2, for the 3, for the 4, for the 5, all the way up in till you reach 23, the ploidy number. Okay. So this isn't going to stimulate any change genetically. Okay. We're not changing anything genetically. There's not going to be any crossing over. There's not going to be any recombination words that, if you haven't heard or remembered, that's coming at the lower part of this page. Okay, so here's an interphase cell. At prophase, the nucleolus disappears. The nuclear membrane begins to disintegrate or disappear. The centrioles migrate towards their poles opposite each other. Okay, and we see that here. And the fourth thing that happens is that the chromatin begins to condense. And now we can begin to see the chromosomes under the microscope. That chromosomal material was there, but in a very loose stage, so that the chromatids were all unwound and able to be read and function. Now they're tightening up, and it's like the store is closed. No more. Not until we get the division underway. Now, this particular diagram used different colors for the father and the mother's set of chromosomes. But why they used four colors beats the hell out of me. It really should be half and half, just two colors. But this is prophase, getting ready. Now at metaphase, I don't know if I used the word a moment ago, the centrioles are fully extended. Their fibers, spindle fibers they're called, are reaching out and touching, grabbing hold of, making contact with, anything you want to say, the centromere. They latch on to the centromere from both sides. But you notice that any chromosome that you look at, one chromatid is the same color as the other side. And that means that the genetic material is the same on both sides, and it's in the same sequence. Nothing is changing here, but now it splits. At anaphase, we have splitting of the centromere. This chromosome here, blue, can tell blue, has been split at the centromere. One chromatid is drawn one way, the other chromatid is drawn the other way. A chromatid loose like that, if you want to think of it that way, is a chromosome because it has its own little piece of centromere. Here we had four centromeres. Once they split, even though they're little halves, we have 8 centromeres. And we have 8 chromosomes suddenly. Out of the 46 that in reality we would have, we actually have 92, 46 going right, 46 going left. That 46 going one way and the 46 going the other way are absolutely genetically identical, unless a mutation has occurred. And we're not talking mutations here. Okay. So what goes right goes to the left. What goes to one cell goes to the other. And they're drawn towards the centrioles. Once those chromatids reach the centriole area, the nuclear membrane reappears, and the cell begins to divide in what is called cytokinesis. Cytokinesis in the animal world is also called furrowing. It's like tightening the belt. And then further, after cytokinesis is complete, we return to having a nucleolus, chromatin, we have the nuclear membrane, and we have the centriole. The centriole can regenerate its missing half, and it'll eventually split when the cell is ready for division. Unless this is a constantly dividing cell, and I don't know of any somatic cells that have to divide constantly. These cells down here will be in interphase, and they will perform their functions. Okay. Questions? Okay. When you take a look at this, you might want to go in the opposite direction than I did. You may want to look at the pictures first and then go up here. Either way, mitosis simply makes more cells, no genetic change. The original cell is diploid, or 2N. The final cells are both diploid, or 2N apiece. Meiosis has two stages, which always seems to make people think it's going to be complicated. It's a little tougher, but we don't, we abbreviate some of these stages. In meiosis, what's the purpose, first off? Down here, the result of two meiotic divisions, because meiosis is a series of two divisions. First cell divides to form two. The two cells that are formed from that divide to form two each, for a total of four. The result of meiosis is to form four haploid cells. Haploid cells have only one set of chromosomes. As it turns out, these will be recombined. They will have shuffled their genetic information and not be the same as their partner cells that were formed at the same time. Each will vary from one cell to the next. The new cells are gametes. So the only place we have meiosis is the formation of sex cells. Sex cells are gametes, but in particular, in the male, they're spermatids, not spermatogonia yet, spermatids. They have to differentiate, change their shape in order to swim, if that's the word, towards the ovum. And in the female, the gamete is the ovum. In the male, one cell located in the testes will divide twice to form four haploid cells. That means that each cell will have 23 chromosomes. Haploid is 23, diploid is 46. These haploid chromosomes will have a genetic makeup unlike any other spermatid in the entire body. It is not conceivable that two spermatids, which will become spermatozoa, will have this genetic information exactly in the same way. The only way we'd get that is with identical twins when the embryo splits. But we're not talking that, we're talking about the formation of the spermatids. Spermatogenesis, jumping to the end here, spermatogenesis is the formation of sperm cells or spermatids, and it happens in men from puberty until death. In the female, the formation of ova is oogenesis. The ovum is large compared to the minuscule spermatid or spermatozoon. The spermatozoon brings to the table only its genetic information. The ovum has the genetic information also, but it has to be larger so that it's got some stored food and the cell can proceed. It needs to have a complement of organelles, such as the mitochondria and the lysosome and the endoplasmic reticulum. The spermatozoan has almost nothing. In order to keep the ovum large, the two divisions of oogenesis, one cell forms two form four, but not equally. The first cell stays large and it splits, and you get one big cell destined to become the ovum, and one tiny little satellite cell called a polar body. The ovum divides again and gives you one more big cell and one more little satellite body, another polar body. In the case of the first polar body, it probably can't survive. But if it does, it may split and form two of the tiniest little cells, still called polar bodies. The polar bodies are not functional. They'll be reabsorbed by the body. The interesting thing most people don't know, some do, oogenesis takes place at birth in the female. Okay, right around birth, a day or two, one side or the other, the number of eggs that the female is going to have for life is determined. Okay, let's do it the other way this time. These are the two divisions: meiosis 1, meiosis 2. I am most interested in meiosis 1. Okay, you have an interphase cell, just like we drew before. But this interphase cell is located in the testes, or it's located in the ovary about the time of a female's birth. Okay, so no change here. The first change that we notice, same thing as prophase was in mitosis. We see the centrioles moving apart, disintegration of the membrane, the chromatin condensing to form the chromosomes, and the nucleolus disappearing. So there's no change here. Okay, we could be setting up for mitosis, but we're not. We're setting up for meiosis. And now we go. Here's metaphase. The centrioles are opposite each other. You have the spindle fibers attaching, but they don't attach to both sides. They attach to one chromosome of the chromosomal pair. In other words, chromosome A from the father or B from the mother will have individual attachments. When we pull on these fishing lines, we will not break centromeres. We will pull an entire chromosome with this one centromere this way and the other one the other way. One thing that this diagram I'm noticing doesn't show you is that in prophase, okay, look at all the material here. The chromatids overlap each other. Oh, you can look at this like a person lying down, hands up, feet out. Cross your legs, one over the other, pull them apart. Okay, and when they pull back apart, they will have exchanged genetic material. Okay, if you think of your hands in front of you, okay, if you're going to cross your hands in front of you, the very first part that's going to cross will be the distal phalanges, where the fingertips are. Then if you pull them back apart, some of them will have exchanged their genetic material from the maternal chromosome to the paternal chromosome. Well, that's just a little change, okay? And the material is always for the same thing. That could be, for instance, for eye color. Now, cross your hands again, cross them to the wrist and pull it back. And now the wrist may interchange from one side to the other. Cross to the forearm, pull it back. They may exchange. What's happening is you're shuffling the chromosomes, and that happens primarily in prophase. Here it says it, crossing over occurs. It's during prophase. The crossing over is like taking a deck of cards and shuffling it. A real quick shuffle, that doesn't do a good job. If you were playing gin or something with somebody, a quick shuffle after the match, no. Let's shuffle it better than that. Shuffle, shuffle, shuffle. The longer you shuffle, the more variation you're going to get. So these chromosomes, okay, there'll be two A chromosomes, two B chromosomes, all the way up to the 23rd letter of our alphabet, but they will all be different. They will all be different. So now we line them up on the equator, and with a spindle fiber pull one chromosome and one centromere this way and one the other way. And the same for this pair of chromosomes. These chromosomes do not belong to your mother or your father. They are a mixture. They are a mixture. Your ancestry has been shuffled here. Okay. At anaphase, the chromosome, and it's still whole, hasn't increased, is pulled towards the spindle fiber. At telophase and cytokinesis, they revert back to this situation right here that we started at. This was a 2N cell at the beginning. 2N cell. 2N cell. This one's split, but the number of chromosomes, half go one way, half go the other. So these are 1N cells. And so our first division gives us two haploid cells. Since these two haploid cells in our diagram are roughly the same size, this must be set up to form spermatids. This could not be set up to form ova, this drawing, because if it were, we would have one large cell and one tiny little cell, okay, the polar body. Okay, so first off, recombination. You could almost write this down if you needed to. Okay, recombination occurs in meiosis 1 and specifically in prophase. That's the shuffling of genetic information. You are not creating any new genetic information, just different combinations of it. And the reduction from 2N to 1N also occurs in meiosis 1. Reduction and recombination both occur in meiosis 1. Move these two cells down at the bottom up to the top on the other side and skip to prophase. Same thing's happening as it did in the other prophase, except we have half the number of chromosomes in each one of these cells. These are haploid cells. haploid cells. We set up the spindle apparatus, and when they pull apart, this time the centromeres are split. Each chromosome is cut in half and has a piece of centromere with it. So what we end up with is 4 cells after cytokinesis here. Well, this is during cytokinesis, actually. We have 4 cells, and we have 1 of every type of chromosome. Okay. So the ploidy number for the human being is 23. Each of these cells has 23 chromosomes, and they darn well do not look genetically like any of the chromosomes that we started out with. They are recombined. They complete their cytokinesis, and we have 4 cells that are haploid, 4 haploid daughter cells. And the chromosomes here very likely have just 1 chromatid, 1 strand, with a part of the centromere, but they are able to regenerate the other half. That does not increase the number of chromosomes. The number of chromosomes is not equal to the number of chromatids. It's equal to the number of centromeres. With all 4 of these might be spermatids, but these have got to develop a neck and a tail and an acrosome at the top, enzymatic adaptation to penetrate the membrane of the ovum. So these need to undergo differentiation to become spermatozoa, and that'll happen in the testes itself. Okay, these are not, though, the final consequences or appearance of 1 large ovum and 3 tiny polar bodies. I think the diagram um, is actually better as long as somebody's explaining it to you. I don't think I need or want to go through these steps. If you do, we have plenty of time to do that. Well, even now, but I'd rather move on to our other stuff. Uh, I will end up giving you some work to do, if you don't mind, over the weekend after the exam to get started on the muscle tissue. Okay, but we won't consider that until after the exam, and I'll send you the information. Any questions? I've been looking at how many people, and I don't expect many. How many people are looking at any of the videos? You know, uh, people who look at the videos, they want to see it again. Didn't get it the first time. Did get it the first time, but don't believe they got it the first time. And then there's the people who never quit. Uh, but the number of views goes up, as we get real close to the exams. Okay, so okay, there we are, 202. Okay, well, if you don't have any questions right now, then we'll take a break and give you a moment to think of a question if you have one, and then we'll go on to the skull. Okay, so it's 203. I cheated us out of a couple of minutes the other day. Uh, Big deal. 2:15. Okay, see ya. Okay, who's here?
Hi, Charlie.
Hey,
Okay, hi. I know I said this in the little email that I probably sent you, but the only thing that you're going to do tomorrow is the practicum. That's at 1 o'clock. You can start as late as 1:30. If you start later than that, you'll probably have already gotten an email from me. When are you going to start? Because you're going to lose time. It'll start subtracting time. Because, well, I need to get the grades in, and I can't do it till I've got everybody. So uh, just help me out. Start no later than 1:30, but 1 o'clock is when it's open. If you've taken one of these tests, you've probably been frustrated sometimes. You click, 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 and the test won't open up, and it says the test doesn't open up till whatever time. Well, it's going to be 1 o'clock. That's when it will open up, and you can start right away. Oh, there are always people that just jump on it, uh, get it over with, and others just sort of wander in. Okay, let's see what else to expect. If you run into a problem during the test, nothing. I mean, not a problem with my um, outline or this or that. I'm talking about a technical problem with the test. Email me right away. We'll see what we can do about it. Okay. Also, looking at your chat, the material that we just covered is nowhere on the test tomorrow. That's lecture material. That'll show up a week from tomorrow, along with everything else that we have done so far and will do next week. So that's why I want you to really jump on the lecture material as quickly as you can after you finish the practicum. Okay. Okay.
I have a question, Charlie.
Sure.
So we were just on the meiosis 1 and 2. I'm kind of confused. Like, where does that happen? Like, I know you said it was the sex organ or the sex cells, but like, where exactly does that happen? Like, when in like conception or before that, or like, I'm kind of confused.
Okay. In the male, the testes are a tubular organ. It's just tunnels and tunnels of tubules. The meiosis begins in the tubular wall. Okay, and then the two divisions proceed towards the lumen. Okay, of this tube and release the final product, which are the spermatids. In the female, it happens in the ovary, and it happens just before birth, the birth of the female.
Okay, got it. Thank you for clarifying.
Okay. I'm going to do something a little bit different. We're still recording, sure, but we are not sharing the screen, right? Good. Don't want to. What I'm going to do, I'm going to black myself out here for a moment. I'll leave my sound on. Okay, I will be here. What I'm going to do is move my camera, and if I didn't black myself out first, I'd be moving the camera, and it would look like there's an earthquake here or something, you know. And I'm going to set it up in front of the skeleton I have. Now I'm going to be doing my best with this skeleton, so you need to bear with me. And also, uh, if you have your book, that's probably going to be quite helpful if you turn to the skull pages. Okay, so I'll be back in a moment, and I'll just lock myself out, like I said, so I can move my camera. Now, when I come back, that's the only time I'm going to know if I move the camera to a good spot. Okay, here we're coming back. That's not a good spot. That's too much light. Oh, I want to pin myself. Okay, as I said, the farther away I get, the more resolution we have. But most of these types of cameras are not that clear. This camera and this damn headset that I have, I'd rather not wear the headset. But when lockdown started and online started a year and a half ago, I went over to Best Buy, and there was no webcams, no webcams at all. I had to get it off of Amazon. And as far as uh, anything else is concerned, really, headsets, no headsets at Best Buy. You believe that? I couldn't. He said everybody's already got them. They all need them for their classes. So I said, "Do you have a gaming set? Because they're more expensive." And yes, they do. So I've got this gaming set that I'll never use for anything. So, okay, so that's why I've got that, and that's why this is a little blurry, but I think we'll make do with it. Okay, I'm going to start right with whatever's in front of me. Okay, this is the alveolar process of the maxilla. The maxilla goes here, up, and into the lower part of the eye. We're going to be doing a little experimenting here on this, I grant you. Wish I had something better to attach this to. Don't like that much light. Okay. This hole right here in the maxilla is the infraorbital foramen. From here up to here, there's a small little suture there that the book will show you. This is the zygomatic bone. The zygomatic bone from here stretches backwards, but it ends right here. And then the temporal bone, or the process of the temporal bone, comes forward, making this structure from two bones the zygomatic arch. Okay. From the zygomatic bone and the temporal bone.
Is it still correct if you refer to it as zygomatic process?
Not really. You're not going to get an opportunity to make that mistake anyway. It's a multiple choice test, okay? And I would not pick two answers so close. The zygomatic process is this structure here. This is the zygomatic process of the temporal bone. This is the temporal process of the zygomatic bone.
Okay.
Underneath here is the external auditory meatus, and the sharp fang-like structure below it is the styloid process. The temporal bone goes from here up and over. I got too much light on it, so okay. This is the temporal bone.
Charlie,
Yeah,
is the external auditory meatus and the external acoustic meatus?
same thing. Same thing.
Okay, thank you.
Okay. So here's the squamous portion of the temporal bone. The rocky or petrous portion includes the external auditory meatus, the styloid process down here, and this larger structure at the back, which is the mastoid process. I'm turning this around because I need certain shadows, and I'm not getting it with this one. Okay. Right in here, you can barely see a line. I realize that some of these are not going to be adequate, and you'll maybe make a note of them and to go to the book. If you were to put your finger right here, okay, behind the zygomatic bone, and aim it straight in. Well, I got to get my hand out of the way. Aim it straight in and above, right here. You would have this area here. This is the sphenoid bone. It's not the only place to see the sphenoid bone. Another place is in the eye, and back here is the sphenoid bone. While we're looking at the eye, so we don't have to do it again, look way back and find the round hole. That's the optic canal. Looking like either a boomerang or a sickle, okay, these cuts here are the superior orbital fissure and the inferior orbital fissure, which allow muscles to enter into the orbit of the eye to move the eye. This is the nasal bones. These are the nasal bones. Looking into the nasal cavity, you see the partition. The lower part of the partition is part of the bone that we call the vomer. The upper part of the partition is the perpendicular plate. That perpendicular plate is of the ethmoid bone. Where does the ethmoid and the vomer part company? Right back in here. And we'll see the vomer from the other side in a moment. We saw the infraorbital foramen. This one does not have a supraorbital foramen. It only has a notch. and then it properly is called a supraorbital notch.
Wait, can you repeat that again? One of them only has the
What?
superorbital notch. Is that the
Supraorbital notch and the supraorbital foramen are both located here. If it's a hole that we find here, it's a foramen. If it's only a jagged cutout, then there is a small piece missing here. It's a notch, supraorbital notch. Okay, what I would like to see next, but I don't know. Let me try the other side. Yeah, there it is. Okay. You see the hole down here in the eye. Well, I can't get it any better. The hole is fine, but it's not on our list. It's a tear duct, and straight up above it is the lacrimal bone. Now, looking underneath, I don't have 3 hands. So how am I gonna do this one? Hang on. There we go. Zygomatic bone, but behind it, this structure right here, coming down. That's one of those 2 feet of the sphenoid bone. That's the pterygoid process. The parietal bone, right here. Here's the coronal suture. If I rotate it, you see it goes how it goes around. There's the mid-sagittal suture. It's coming straight back. Here's the frontal bone, parietal bone. Okay, now I'm going to take the lid off on this guy. And first, start by turning him over. The occipital bone comes up from the posterior side. It surrounds the foramen magnum. Then these projections here and here are the occipital condyles. The external occipital protuberance is located back here. Here's the hard palate. It's the palatine process of the maxilla. If we're looking for the palatine bone, it's located back here. And there's no good separation, even if this was a perfect shot. But we can tell the palatine bone usually that we've got it because there's usually small little foramina. One right there. Okay, can you see that one? I can't, yeah, I can do it. It's right here. The foramina is always in the middle of the palatine bone, which is here. Vertical, this bone separating the two halves of the nasal cavity, this is the back end of what I showed you from the front. Okay, this is entirely the vomer. This is the vomer. On either side of the vomer, these two trough-like structures, these are the pterygoid processes again. If I show it to you from this angle back here, that's always vomer. There's no perpendicular plate back here.
Charlie, can you point to the pterygoid processes again?
Here and here.
Okay, thank you.
Okay. Okay, now looking through this might be actually better. This hole and the one on the other side of it are the foramen lacerum. This one here is the foramen ovale, and the foramen ovale on the other side. The foramen spinosum seems to be filled in. That's the trouble with plastic models. They're small. They would make the heel of this structure. Also, this structure here and on the other side here, these are the mandibular fossa, the mandibular process of the mandible would fit into those. Okay, once again, foramen lacerum, here, foramen ovale, and there is a tiny opening here for the foramen spinosum. Frontal bone is here, and sticking up through it is the ethmoid bone. Let me try do it by hand. Yeah, it's better. Here's the ethmoid bone. On either side of that are a number of holes. They're called olfactory foramina or cribriform foramina. They go through the cribriform plates, like Swiss cheese. The cheese itself would be the cribriform plate, and the hole going through the Swiss cheese is not a structure. It doesn't exist. I mean, you can't put a pin on it, but that would be the olfactory foramina. This is the sphenoid bone, and this part of it is the lesser wing. The greater wing is down here. The saddle is the sella turcica. That's where the pituitary gland fits. And in this, we can find a small opening here and here, and they're headed out to the eyes. Those are off the optic canals that we saw from the other side. From this side, much better. Foramen spinosum, foramen, I'm sorry, I said it wrong. Foramen ovale, foramen spinosum, and up here, the foramen rotundum. This is part of the petrous portion of the temporal bone. This large opening is the jugular foramen. Jugular foramen. Right above it, and only visible from the underside, like or the upper side, like this. This little hole opening here, or over here. That's the internal auditory meatus. Internal auditory meatus. You cannot see the opening to the carotid canal from this side. I need to turn it back over again. And that is right here. It's not dark because it doesn't go straight through. It actually turns sideways and goes medially. That's the carotid canal. When you want to see the jugular foramen, it's quite visible right here.
I have a question.
Yes.
So I have the carotid foramen also written down as the foramen ovale. Do I have them like do they have two names or am I
No, they're entirely different.
okay?
Okay. Let me do the carotid foramen first, okay?
Okay.
And it's right here. And you don't see it going through because it turns sideways. That's the carotid foramen. And there'd be one of those on the other side as well, right here. The foramen ovale can be seen from this side. It's right here.
Oh, okay.
Okay, it's up there. And here's the foramen lacerum. The carotid canal cannot be seen from the upper side. Okay, so we don't look for it.
Okay.
But here's a really nice foramen spinosum. I did it again. I guess I'm going to miss that one tomorrow. Foramen ovale. Behind it, the foramen spinosum.
Okay. Thank you.
Okay.
Charlie, can you point out the optic canal again?
Let me do it through the eye. Look back and medial in the eye, and you find a round hole. That's it. Right there. I'm covering it right now. Now it's
Thank you.
open.
Okay.
Okay. Okay, I'm gonna get the mandible because we didn't do that. I'm going to black us out for just a second. I'm still looking.
Okay.
Okay, are you back to seeing cell division?
Yes.
Okay. Uh, for some reason, I don't find the mandible, so I'll just get a good picture. Mandible's the easy one. No, 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 no. Okay, I'm using this one because it doesn't have any labels. Labels are nice, but well, maybe that's a better color. Okay, on the mandible, the body, if you can put your chin in your hand, put like a horseshoe shape, that's the body going all the way around. The alveolar process holds the teeth. The mental foramen is here. The mandibular foramen is not showing on this picture, should be here. I'll find another picture where it is in a second. Let's just continue with this guy. This is the angle. This is the ramus. Back here is the mandibular process. It has other names, but we're using mandibular process, fits into the mandibular fossa. This is the coronoid process. I don't know if that's a nail, but it's in the exact spot of the
Charlie,
yeah.
in our book, I think those are kind of reversed. Like in the book, the coronoid process is the one that's closest to the teeth, and the
That's,
condylar process is the one behind.
I'm sure that's exactly what I showed. The coronoid is here.
Oh, okay.
The mandibular is back here. Yeah.
Where's the condylar process?
The condylar process is also the same as the mandibular process.
Got it. Okay.
Another name for it. Yeah, that's something that you just got to take a deep breath on and realize there's more than one name for almost everything that's in the book, it seems. And you wonder, why did they do that? What, if you wanted to be precise, let's just have one language. But they don't. Okay, you can't convince everybody, it seems. Okay, but that's the condylar process. That's also the mandibular process. Over in this side, if you can see it, they're calling that the condyle. And here's the coronoid process. That's right. If they want to call that condyle, they better call it condylar process. Okay. I'm not going to put any ringers in the test that should fool you. For first thing, even though there may be two words for something or two answers for something, only one of them is going to be provided on the test. So you got to give me the one that's there. Here's condyle again. Old book, old book. Okay. Well, the things that give people the greatest amount of trouble are not these things here on the mandible. And okay. The things that give people the greatest amount of trouble: the perpendicular plate versus the vomer. I think this one's right out of our book. It certainly looks familiar. The vomer is down at the bottom when we're looking at it from the front here. And the perpendicular plate sticks down from it's the lower end of the ethmoid. The higher end of the ethmoid is the crista galli. And I didn't show you this when I had the skull out. We need to look at the ethmoid bone inside the orbit of the eye. Oh, oh, shut up. That's all I needed. Hit one of those things. Okay, so uh, okay, here we are with the orbit of the eye. Here's the easy starting point, I think. The zygomatic bone. See how it creeps into the orbit? right in here. Move over to the maxilla, and in the same color, even I can tell it. This is the maxilla. Take the frontal bone, and it makes up the top portion of the eye. The sphenoid bone that we looked at on the outside and then looked at inside the orbit. Here's the greater wing of the sphenoid bone, making up the back or posterior part of the orbit. The ethmoid bone is here. I don't know the color, but you can see it right there, coming forward to the lacrimal bone. Okay. That's 6 of the 7 bones, and the 7th bone that makes up the orbit is the palatine bone. But it's such a small piece right here, right there, that I can't show it to you, so I don't ask you about it.
Charlie,
Yes.
looking at different photographs of the skull, and it's kind of hard to differentiate the lacrimal and the ethmoid bone in those pictures, since they're right in front and behind of each other in
Yes.
the photographs. Is
Yeah.
there an easy way to tell?
Let me take a look around myself here, okay?
Okay.
You can always tell these things when they have color coding. That's really not fair. We don't know if this will enlarge. Come on. There it is. What the hell is that? I got myself into the dark web there. I know exactly what I'm looking for to show you. In this one, here's the lacrimal bone, and you would know it because that hollow area, that duct, is right underneath it. Okay.
Okay.
And then immediately behind it, all the way to the back, is the ethmoid. The ethmoid is actually quite smooth, and that's what I wanted to have you look at. No. Okay. You never get what you want. No. Different vocabulary. Here's one. But again, this is a drawing. Okay. Here's the lacrimal bone right here. And right behind it, the rest of the way, nice and smooth, is the ethmoid bone.
I see. So the angles at which they protrude kind of differentiate them.
That's one way, sure. That's one way. If you had an actual skull in front of you, let's see. I'm taking a look at the skull I have, and it wouldn't help you. That's the trouble again with plastic.
Thanks, Charlie.
Here's one more. Okay, again, it's here's the lacrimal bone. It always is surrounding the canal, and then behind it, you see a fissure or a suture, I should call it. Okay, and behind that is the ethmoid bone. Okay. And, there's a lot of people here, and I assume a lot of people are waiting for somebody else to ask a question that they can benefit from. But if there aren't questions, well, then we're taking away from study time. So I'm going to stay until I've got the last question, but I don't really see anything more enlightening coming. If you do, then you should be asking a question. Okay.
Charlie, can we ask about other parts of the body now, or is it only the skull still?
Oh, anything now. Anything.
Can you point out the subscapular fossa?
Okay.
Or is it just the whole center part?
It's the entire center part, but the anterior part, and it's quite smooth.
Okay.
It looks like an ashtray.
Okay. Thank you.
Okay, well, here's a picture of it anyway. It's right here.
Can't see your screen.
Oh, darn. I took it off of that, didn't I? Okay. Here I am. I'm back. Okay. Oh, here we go. Okay, now can you see it?
Yes, I can.
That's the subscapular.
Okay.
Charlie, is that also considered the body?
No, not of the scapula. No.
Okay.
Okay.
You said we're free to go, Charlie, and study?
Oh yeah.
Okay. Thank you.
Yep.
I had a question. You mentioned that will you be sending us an email after tomorrow with what to start studying for next week? Is that what you said, or?
Yeah, but I can tell you right now what you need to start with, and that's very simple. The notes that I gave you, if we had been in class, I would have put them on the board. Okay, instead, they're typed out for you. Okay, and it's every item in the modules that says lecture after it that we did so far. Okay, that's a good place to get started. Okay, and now I'm serious here. You need, those notes are not big volumes, but you need to memorize them. You need to be able to recite them forward and backward. Okay, that's if you're looking to get every question right. I don't make hellacious lecture tests. Nobody, I don't think anybody believes that. But I want you to realize it will be harder than the test tomorrow. So you gotta put some effort into it, okay? Or just study when you're clear-headed, absolute clear-headed. Some people study in the morning and they're not awake. Some people at other times of the day, or they're exhausted, or they're just getting home from work. You got to find that time to study that. You read it and it really sinks in. Okay.
Charlie.
Thank you.
Yeah.
And it probably goes without saying that there's not going to be as many pictures on the other
No,
practicum.
just a couple of pictures. Okay.
Okay.
You'll be ready for them. Don't worry about that. But no, there's going to be 100 pictures tomorrow. Please check your internet. Tell the other people at your house you're going to take a test at 1 o'clock. It's only going to last an hour and 20 minutes. Please don't be using the internet or Wi-Fi if possible. Otherwise, you may dilute down your signal and we run into problems. Okay.
I have a question.
Okay.
When we were going over the ossification lecture, you pointed to an image of like the Haversian system and said that it would be on the practicum. Is that true?
One question only. One question is, there were going to be 5 more, but those are the 5 that I eliminated. Okay, I told you it was set for 105 questions, and it should be set for 100 questions and 100 points. So I had to take some things out. I left one only.
Okay. Thank you.
Okay.
See you, Charlie.
Okay.
I thank you.
Thank you.